So we are on our last part of culturing, quote-unquote, microbes. So how do we, the ways we can identify which microbes we have. And on Tuesday we talked about immunological methods. I want to remind you that they're based on the interaction between the antigen and antibody, right? And we did talk about the Western blot, how you separate the proteins that you have in your sample based on their mass when they move in the electric field, essentially, and then you add antibody, which will bind only to the desired protein. One thing that you will see in common in many methods is that you find what you look for. You probably know that phenomenon when you look for something at home. Like you look for the keys, and then your spouse asks you, "Did you see my purse?" You have no freaking idea because you didn't look for the purse. So you may have seen it a million times when you were looking for the keys, but you're not aware. Same here. You will find the protein that you have antibodies to. You have thousands of them, but they all go undetected. Does that make sense? And same goes for anything else, like ELISA. That's the thing. You know, you have a patient. That's the problem. Problem with the clinical diagnostics. You have a patient that walks in. Well, if patient walks in with a cough, you're going to tell them, "Go home, tough it out," and that's about it. But then patient comes with something more serious, and you start running tests, and you think, "Okay, that's syphilis." You run the test. No, it's not syphilis. You run, oh, it's gonorrhea. No, it's not gonorrhea. So start running many tests looking for one thing at a time. Okay, and there are methods that try to overcome that limitation, that try to look at everything. So same goes, well, immunofluorescence is somewhat better because think about this. You may have patient's tissue sample, and now you can have antibodies that have all kinds of wonderful colors. Honestly, it's usually yellow, green, and red, but it's already 3 instead of 1, so like 3 times more. So you run the test, you stain, you see. If you have cells that are colored that bind your antibodies, they're probably infected with whatever these antibodies recognize. Okay, so that's immunofluorescence is really a good thing for diagnostic purposes. Now this is, I listed them under the biochemical methods, and I will tell you why. But essentially, the concept is more about physics, and I understand that this word physics can cause students to drop dead in the middle of a class. But I will try to look. Technicalities behind these methods are huge, like really huge. If you would look at the chromatography setup, it's like a lot of hoses and pipes and just like a plumbing system multiplied by 100. If you would look at the mass spec, it's a gigantic box with a lot of stuff inside which only certified technicians would know what to do. But basic concepts are pretty similar. And, well, similar to an extent. They're not very complicated. So here's the idea. Microbes produce huge number of chemicals, right? I mean, they metabolize things and produce chemicals. What you can do, you can take the microbial culture and run it through the column. Now, what is the chromatography? Is the part of analytical chemistry that studies how different chemicals behave on different sorts of absorbent materials. Imagine that at home, you take a piece of paper, like a construction paper, and you stick it into water. What will water do? Yeah, it will go up, right, through the paper. Imagine you add several water-soluble dyes, like watercolors, into the water. What will watercolors do? Yeah, they will stay in the paper. Now, it depends on what the size of dye molecules are. Some of them may move slightly faster, some of them may move slightly slower, right? So you may end up with the mixture of watercolors being separated on the paper. That make sense? This is chromatography. This is just one type of chromatography. Big machines, the fast machines, they use columns, which operate on a similar principle, but in this case, the solution goes down. It can either go because of the gravity, which will take forever, or usually you may have a pump. This pump just pushes it through. Does that make sense? Some compounds will go faster and some compounds will go slower. Does that make sense? What can determine which ones are faster, which ones are slower? What do you think? Size. Size, very good. Size. Now the sorbent, the thing that fills up the tube, is a chemical. So what else? The charge can. Excellent. So chemical nature, charge. There are many, so three major things: size of the molecule, charge of the molecule, and chemical nature. If it's water soluble or lipid soluble or something like that. Does that make sense? All of that will define which compound will leave the column first and which compound will leave the column last. You can detect when they get out using different types of detectors right here. You can collect fractions, you can analyze fractions afterwards. Does that make sense? Or you can just say, I have a standard, I'm going to run the standard first, I'm going to have some sort of a graph like this, okay, with peaks corresponding to different compounds, and then I'm going to throw in whatever sample I have, run it through as well, and compare the standard and my sample and see what's going on. Does that make sense? So that's chromatography. It's fairly expensive. Setup is expensive, but once you buy it, it serves for years. So it's convenient. Mass spectrometry is a very interesting method that for a long time was used only by scientists, not by clinicians. And now it makes its way into the clinical diagnostic labs. This method also deals with a separation. This time, not so much separation of molecules, but separation of the fragments of molecules. What you do is here, you inject your sample in. Now, remember I told you about the word fancy that I use? It's very complex and very expensive. It's fancy in both terms. It's very fancy. So inject your sample, and then through a series of magic tricks, molecules in a sample become ions, which means what happens to them? They gain positive or negatively. So they gain charge. okay, and they are shot. I mean, not like they shot dead, they shot in the tube. Molecules have different charge, and they also have different mass. Does that make sense? Which ones are going to fly farther? Smaller. So first of all, you can separate them by mass. The magnet here will change the trajectory of the flying molecules based on the charge. Make sense? So you have two factors, charge and mass. It turns out that for the method is designed in a way that for pretty much every molecule, the charge and mass are going to be unique. So you can identify the specific fragment of a molecule or a molecule based on where it is, where it ends up in that mass spectrometer. Does that make sense? So it will produce the graph like that with peaks corresponding to different molecules, and the height of the peak will correspond to how many of those molecules you detected. By itself, this graph is honest. I mean, it is useful for a scientist, but for clinicians do not have days to go through piles of data using software to identify something. What is done instead? You take, say, E. coli. You prepare the sample. You put it in the machine. And in the end, software gives you a graph. That's going to be your unique signature for this strain of this bacteria. Does that make sense? Next time you have a patient with some sort of an infection, I don't know, urinary tract infection, you collect the sample, you do the culture, you subculture it, you make pure culture, you collect bacteria that you don't know anything about yet, stick it into the chamber, run it, get the experimental graph, compare it to the signature. If they match, you got it. Does that make sense? Of course, it's not every time. Now we don't even have to run the control sample. We have databases. So you can compare your experimental sample with a database, and it will tell you what it's most likely, okay? Whether it's E. coli or salmonella or something else. That is really awesome. When you have a sample, this whole thing takes about 2 hours. It's really fast. The downside is that you have to have it in the form of a pure culture. You still have to subculture it. Second downside, what do you think? $750,000 for the machine. Not every hospital in the United States can afford it. And hospitals are not the poorest enterprises here, but not everyone can afford that. There may be tens of those machines in states in the biggest centers. Yeah, that's pretty crazy. Now, question? No? Now the next, and pretty much the last method for identification, is genetic. Well, when I say genetic, it means that we're dealing with the DNA. DNA, or in some cases RNA. Well, we're going to chat about DNA here. How many of you have heard of PCR, polymerase chain reaction? Ooh, good, awesome, terrific. Polymerase chain reaction. Forgot the name of the guy, Carey. I know his name is Carey. The last name starts with M. They got he and his buddy got Nobel Prize for developing this system. It is based on the property of the DNA, okay, to get copied. The basic idea behind PCR is that all the species of everything, starting from viruses and ending with humans, have unique sequences, unique parts of the DNA that can be identified. Does that make sense? Even we, all of us, we do have unique parts of the DNA. So some part of my DNA would be different from Chris's DNA, okay? And this technique allows us to specifically amplify and increase the number of copies of that particular part. Okay, we're going to talk how we do that, and then what conclusion we can make from that. Does that make sense? So how we do that? Imagine that this green stuff is the part of DNA that you would like to detect. You do not know if it is present in a sample or not. For the purpose of explanation, we assume that this part of DNA is in the sample. Make sense? And DNA comes from the cells, right? So let's say you have 1,000 cells of bacteria. Obviously, you cannot, by any means, visualize 1,000 DNA molecules in the lab. I'm not even speaking to find that one unique piece in there. Does that make sense? So you have to come up with something else. So this is PCR. First, and those species, it's double strand, okay? We cannot do squat with double strand, because the nitrogen bases, they pair with each other. They're not exposed, just isolated from reading, like hidden. So first we need to split them apart. These nitrogen bases are interacting by hydrogen bonds. What is the easiest way to break down hydrogen bonds? How? Heat. Yeah. We almost boil it. DNA is pretty tough, so it's okay. We denature it. You see? So strands become separated. If you're not following me, if something is unclear, please let me know. Okay? So far, so good. Next, we add special molecules, fragments of DNA that are pre-synthesized. These fragments are called primers. This red fragments. These are not random sequences. Primers are complementary. As you can see here, to the beginnings of the fragment that we want to detect. Does that make sense? So we use primers to kind of restrict the part of DNA that we're going to work with. Does that make sense? So essentially, primers tell to the enzyme, you're going to start from here and from here. That's the starting point. Does that make sense? Got it? It's a starting point for the enzyme. Enzyme is not going to go somewhere like here or somewhere here. It's going to start from that point. Enzyme needs primers. It cannot work without them. It needs a starting point. It must be told. Next, enzyme, which is DNA polymerase. I'm going to picture enzyme like this. DNA polymerase. It starts to make a new strand. You can see it in the blue color. Okay? Now, obviously this strand has no restriction at the end. It will go as far as it can. Okay? What's going to happen next? That's first cycle. During second cycle, the whole thing, starting from denaturation and blah, 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 will go on. Now, here's what's going to happen. see this? This molecule right here, the newly synthesized, will be complementary to newly synthesized molecule right here. Does that make sense? They will be complementary. They will bind to each other. And they will self-restrict. So eventually, what you're going to have fragments of DNA that are of exact length. And that length is defined by the places your primers bind to. Okay? Let's pass on technicalities and talk about the major outcome. Essentially, what you do, going back, making analogy to the cookbook, remember? The cookbook that I told you about. So I have 1,000-page cookbook. And somebody asked you, or your friends asked you, to get a recipe that starts with the word grandmother and ends with the word fox. I don't know why grandmother and fox in the recipe book, just first that came up to my mind. Okay? So you find those two words and you copy whatever's in between them. You make 1,000 copies. That makes sense? Now, going back to actual PCR. If you start with one molecule of DNA, after one cycle, how many molecules are you going to have? Show. 2. After 2 cycles. 4. After 3 cycles. 3, no. After 4, they double. After 20, 1,048,576. Okay, pretty much with every 10 cycles, you increase the number of molecules of DNA 1,000 times. So if you start with one, that one unique fragment, after say 40 cycles, you're going to have trillion copies of that fragment. What you can do then, you can take the sample where the reaction goes and put it on, detect those fragments. Let's not, again, go in the details. Okay, you can detect specifically that little fragment that you multiplied. Does that make sense? So you start with, say, one huge molecule of DNA. Using primers, you select one little piece of it and make trillion copies. And then you can see those trillion copies. Does that make sense? Why do we do it? If you select something that is absolutely unique for, say, influenza virus, something, some sequence, some part of DNA, well, in case of, okay, DNA, whatever, well, it's not a part of genome that can be found only in influenza virus and nowhere else, you can very reliably detect influenza in a sample of the patient. Does that make sense? You got it? So you look for something, and it's extremely specific, and it's extremely sensitive. Technically speaking, current modified PCR methods may let you to detect one DNA molecule. Physically, one. If you have one DNA molecule of virus in the sample, you can find it. It's not going to be easy, but you can find it. This is pretty neat. Does that make sense? Another big advantage is it can be automated fairly easily, and it's relatively fast. The whole DNA, the whole PCR thing with the detection step can be about 2-3 hours. Okay, that's cool. And you can actually, well, DNA is not just floating around, so you can automate the part of DNA isolation. And I saw these machines, and they're awesome. Well, overall it's about $100,000, the whole system. You pretty much put several machines one next to each other, and all you need to do is just, when one step is done, you put tubes in another machine. That's all you do. They already have all reagents, so you load all reagents, you push the button, they go. You move to another machine, you push the button, and it goes. So it's high throughput method. It allows you to analyze a huge amount of samples in a fairly short period of time. Okay, and it's relatively cheap. So we talk about several dollars per sample. Yes?
Would you analyze there's a virus or the individual's DNA that might have been modified by the virus?
Whatever you want. You can do whatever you want with it. If you want to look at the distribution of a gene in the population, I don't know, one thing that comes up to my mind, alcohol dehydrogenase. It's a gene that is responsible for breakdown of alcohol. The people with Asian ethnicity, Chinese, Japanese, American Indians, they have very low levels of that gene. Caucasians have high levels of the gene. You can see the distribution of the gene across population. For example, how many copies do you have? Something like that. So it's really, it's really great method. You can, like, say you do survey of, have you donated blood? Some of you at least. I bet you have. One of the things that they tell you what your blood is going to be tested for, HIV. That's how they do it. They just stick your blood in the sequence of machines, and it runs very quickly, detects at a very extremely high sensitivity. So you can find several copies of viral genome in your sample. That makes sense? You can do this. You have, say, there's an E. coli strain called H7O157. It causes enterohemorrhagic pathology, really bad, hemolytic uremic syndrome, really, really bad. People die from it sometimes, really bad. You can do a survey. How many people have it? Find specific part of E. coli genome of that strain that is unique for this strain, collect stool samples, isolate DNA, run. It will recognize only this part. So, I mean, you don't even have to like separate in subculture. It's really neat. Does that make sense? But the problem is, again, that you find only what you're looking for. So you don't know what else do you have. And here we have sequencing. Do you know who Arthur Clarke is? Well, new generation. Arthur Clarke was British sci-fi writer. Great guy. Really, I highly recommend his books, especially the short story named 9 Billion Names of a God. Something like that. It's hilarious story. Very short, very grim, very funny. Once Clarke said that very highly advanced technology cannot be distinguished from magic. And with sequencing, at least for me, it is true. I have absolutely zero clue how it works. I mean, I understand how this works. Not in the details, but I have a fair amount of idea. We all understand how this works, you know. We have an idea, general idea how car works. Engine, wheels, transmission, all that stuff. Sequencing, I absolutely honestly, I know that they put in the sample and get the results. What's in between? It's magic, really. So what did I tried, and I couldn't. I'm not smart enough for that. So what it does, and that's what we want to focus on. Each genome has a sequence of nucleotides, right? It's a chain of nucleotides. It can be 3 billion like in humans, or it can be, I don't know, 1800 like in hepatitis D virus. Sequencing allows you to read this whole chain, to know what's in there. Like we know now all nucleotides of the human genome. We know the whole sequence, right? It took us something like 4 or 5 years and $200 million to sequence the whole human genome. Last week, Illumina, I maybe told you this. Illumina, it's a biotechnology company, announced they're gonna release a special machine on the market. It will sequence the entire human genome under $100 in several hours. So come into a doctor, they collect the blood sample, stick in the machine, and they have your genome right away. That's just mind-boggling. It's like it took 15 years, about 15 years for that evolution. So sequencing, why sequencing is so awesome. And we'll talk about downsides. Now, imagine you have a patient, and patient comes over and has some horrible disease. So you decide, I'm going to sequence everything. So take the blood sample, stool sample, stick it, well, not mixing it, but sticking it separately in the machine, and you get everything that's in a blood sample. You do have databases of gene sequences, and you figure out that, well, human genes you're not so much interested in, that this patient has metapneumovirus, herpes, CMV, staph, and gonorrhea in his blood sample. And in a stool sample, it's a shitload of microbes, like 1,500 species. Some of them are more abundant, some less abundant, some common, some uncommon. You can do it. It's not a problem. What's the problem? What's next? Huh? It's going to be a pain in the ass, right? Because you have so many things. What is that? What is causing a problem? That makes sense? Like having too little information and having too much information, it's both bad. You see what I try to say here? Now, I kind of blew off the whole idea of sequencing being useful. Turns out, can be. There was a very interesting case. A 2-year-old girl, I think it was, well, don't remember the exact age. She went down with the flu. It was not just regular flu. She was that close to be dead. And nobody understood why. Because otherwise, before flu, she seemed absolutely normal. All tests were fine. She was a healthy kid. Well, physicians decided to sequence the genome. It was about a couple of years ago, so it took them a couple of days. They got the sequence. It turned out she didn't have one protein of the immune system, certain immune pathway, just one, which made her extremely susceptible to influenza. She got really, really sick of it. Make sense? Otherwise, they wouldn't understand what's going on. So at least they knew that's the problem is in the immunity, and they started to act accordingly. And they actually cured her. And now she knows that she has a certain immune deficiency that does not manifest in any other way. So when she recovered, she's absolutely fine. There's no phenotype whatsoever. So that may come in handy. That's why I'm so excited about that Illumina thing. To know what you have, what you don't have, because certain things will never show up. Does that make sense? Any questions? Now, it seems a little bit overwhelming, so I will kind of tell you what you're supposed to know. For this guy, for sequencing, understand what the result is. I'm not asking for technique. So what does sequencing produce? What does it show us? Okay? The lineup of nucleotides in the genome, pretty much. Be able to explain the disadvantages and advantages actually of every method. Okay? Again, I'm not talking about a lot of technicalities from the clinical standpoint, finding only what we look for on one side and having a ton of potential suspects and not knowing which one is the actual problem. Question of the methods that we discussed. We have discussed 7. So again, major advantage of methods like ELISA or PCR is that they fairly inexpensive and they're fairly fast. Disadvantage, we find only what we look for. Okay. Sequence is a little bit more expensive, but we have a ton of information. Does that make sense? For PCR, basic idea, you amplify DNA, increasing the signal, and then you detect the signal. Okay, so you look for certain genomic information. For this stuff, creating molecular signatures. In case of chromatography, you create specific molecular signature, and in case of mass spec, you create a molecular signature. Now, this is the method when you can just stick your microbe, run it through, and compare the results to database, and you get what is there. But again, if you have 5 different microbes, you have to do it for each and every one of them. Does that make sense? When you have 5 types of colonies, you run those 5 colonies, and then you look. Okay? And this one, again, convenience, it's very well developed. It allows you to detect the actual antigen of the actual cell fragment. Downside, you find what you look for. Right? There was something I wanted to mention and I completely forgot. Well, maybe it'll pop up later. I think it was something about this. I don't remember. Sorry, I just something. If I didn't say it, no worries about it. Okay. Oh, it's more of a, remembered what I wanted to say. When I say you get a signature, okay, every time you do a clinical diagnostic thing, remember, clinical lab will not tell you your patient is sick because of such and such microbe. Does that make sense? They will only tell you your patient's sample has such and such microbes. And it's your judgment what's wrong with the patient. Does that make sense? It's not a job for clinical lab to give a diagnosis. Question? No? No questions yet. Okay, good. We're going to start chatting about the cell today. We talked about spontaneous generation. Remind you, Redi, this guy, Francesco Redi, covered the meat with, if you look at different sources, some say it's a quark, some say it's a cloth, okay? no maggots. Sorry. So if meat is covered, no maggots. The first blow, and that is the famous Pasteur experiment that I told you about. The S-shaped, or whatever you want to say, U-shaped neck or the flask, when it stays intact, the media remains sterile. When it's broken right here, the media develops bacterial growth. Okay. Now, we have a privilege to know a lot of stuff about cells, and that they are the building block of life. Reddy did not know that. Pasteur might have, because at the Pasteur time, these four fellas, Schleiden, Schwann, Remarque, and Virchow, they established the idea that cell is actually a biological unit, elementary biological unit, something that can reproduce, something that can give rise to other cells, either in the form of like just division, mitosis, or in the form of differentiation. When you have one cell that differentiates into another cell. Okay, so that was very important. Hooke was the first to observe them, but he never made the link. So you see, it's very important to make a link. We'll get back to it. How what is the difference between observation and the discovery? You may observe things, but you may never discover the effect. But then you observe things, you link two things together, you understand, oh, this may have such implications, or this was caused by this. And then you make a discovery. Okay? That makes sense? I'm going to talk about germ theory of disease because we kind of covered it already. Just to remind you, bless you, that Semmelweis and Lister can be, listed as major contributors to the developing the antiseptic techniques. The side effect of their observations was that understanding that infections, that diseases are caused by something, diseases that can be caused by something that's infectious, that's something that is transmittable. Make sense? Like in case of Semmelweis, when he found that his residents transmitted microbes from morgue to patients in the maternity ward. So transmission on the hands. Okay, that was a big contribution. And finally, Pasteur and Koch developed the germ theory of disease. Pasteur suggested that microbes, he actually called all microbes viruses. He coined the term virus, but not in the sense that we know it now. He suggested that microbes can cause the disease, and Koch should explain how we can link. Actually, I'd rather talk about it a little now because we're going to go back to Koch's postulates again. So you have a patient who is sick, and how can and you suspect that the patient has patient has diarrhea and vomiting, and you think that patient has a norovirus infection. How can you confirm that norovirus indeed is something that causes the infection? What should you start with? Okay, so you have to isolate it first, right? So you have to isolate the pathogen, preferably pure. Okay, you got your pathogen. What do you do next? I mean, if patient has norovirus, it doesn't mean that norovirus causes it. You may have a load of other microbes, potentially pathogenic microbes, and they not necessarily cause the disease. So what do you do next?
Did you say you collected it from the urine?
No, norovirus is fecal, but it is you have your suspect. How can you prove suspect guilty? That's Dr. Howe's approach. There is no treatment for norovirus, so we're basically screwed. Bigger. Think more general. So what if you have a sick patient and you have norovirus isolated from sick patient? How can you prove yes, causative relationship? What? You're gonna inoculate what? Forget about ethics. Excellent. So you put it in the model. In some cases, it's only humans. Okay. Generally, since we can't forget about ethics, we put it in the animal. Okay. So we put it in the animal. What's the next step? Exactly. You observe. What should happen to the animal? Excellent. So it not only has to get the disease, it has to get the same disease. It's kind of a problem. Mice don't puke. They don't. They just don't. They don't puke. They do have diarrhea, but yes.
Didn't mean to interrupt you, but
Go ahead.
I thought that a lot of like viruses
Mhm.
and bacteria they don't cross over.
To species? Absolutely true. Great observation. A lot of pathogens are entirely species specific. Entirely. And that's the problem. Okay? Because Koch was formulating postulates when we knew much less. So it turns out Koch's postulates almost never work because there are so many limitations. So they're more theoretical. But okay, so far, got patient, got the microbe, put the microbe in the model got sick, and if we're lucky, model developed the same sign of same disease. Okay? What we forgot to do first when we isolated the sample from sick patient? What did we forget to do? No, we cultured it. That's how we isolated. We need to collect something else, some other sample. Say again? Say it louder. Okay, so we need to inoculate a good, healthy animal. What else? In the very beginning, we collect, yes? What do we have? So we collected a sample from the sick patient. Yes, we need one from the healthy patient, and we have to make sure that sample from healthy patient does not have whatever we suspect. Does that make sense? Okay, so we got those samples, we cleared our controls, we have animal that is sick. What do you think we're going to do next? At this point, it doesn't really matter. Oh, we're not talking about treatment. Koch didn't care about treatment. He cared about diagnostics. Does that make sense? Huh? So, I mean, it's a good idea about vaccine, but he didn't care about vaccine. To see what? Excellent. To see if that same suspect is in, say, in this case, norovirus is in the mouse feces. And if it's there, then we're good. Does that make sense? So that's Koch's postulates. And as we just saw, they never work. There's no animal model, they don't do the same damage to animals, and blah, blah, blah. Okay, so it's really hard. It's really hard. So there are troubles linking. Okay, next topic I want to chat. Endosymbiosis. So we have pro and eukaryotes. Okay. Eukaryotes have nucleus and organelles. Prokaryotes don't. First of all, it's kind of a depressing. We have no idea how nucleus formed. Really. Somehow. We don't really know. It just formed. Eukaryotes are not very much younger than prokaryotes. But someday it just formed. Okay, we got the envelope around the DNA. Okay? But what happened next is that eukaryotes acquired mitochondria and chloroplasts. The guy Mereshkovsky first noticed similarities between the chloroplasts and bacteria, and then Wallin saw the same for mitochondria. What are the similarities? Can you tell me what is similar between, say, mitochondria and bacteria? Anyone? Well, okay, bacteria have metabolism, but it does not necessarily mean that they are related. First of all, mitochondria has DNA, and chloroplasts do have DNA. So they have their own genetic material that is different from cellular. That's one. Second, can you reproduce mitochondrial chloroplasts independently without the cell? You can put chloroplasts in the egg, and they will divide in the egg. They divide. They have the capacity of division, not like independent living, but they can divide. So that was Lynn Margulis who suggested that those organelles, the mitochondria and chloroplasts, so you can see mitochondria and chloroplasts, were acquired as endosymbionts. Endo, what does that mean? Inside. Endo means inside. Symbiont, something that lives together with the host. So it's something that lives in the host. What was the advantage of that endosymbiosis for the host, for the first very, very ancient eukaryote? Awesome. Yes, the cell, the host, got the energy. Chloroplasts or mitochondria got physical protection, and in some cases, it expanded the ecological niches, maybe added some biochemistry to availability of food, nutrients, stuff like that. Okay, so they both benefited. And that's it. Actually, we see some examples of endosymbiosis now. There's one system where you have tick protozoa and the bacteria inside of the protozoa. And all three of them, they can't really survive independently. So they're really protozoa and bacteria endosymbionts. So we still see that process going on. It just takes way longer than we can observe. So what is the span of recorded human history, like, realistically? 5,000 years? 10,000 years? It's a blink of an eye in terms of evolution. In evolution, we talk millions. We chat about structure, then we'll get to our results, then we take a break. Structure. I'm really proud of that thing. I didn't picture it, by the way. It's not mine. But I did those little red things. You know? The writings. It gives you an idea. You must know the main function of those things. And some of you, none of you actually are familiar with my new favorite saying. How do we eat an elephant? How do we eat an elephant? Yes, one bite at a time, piece by piece. Okay? So same goes for all your studying. Don't try to study everything. Study piece by piece. Same goes for the cell structure. So imagine a cell. What does it have to have? All cells. No exceptions. DNA. What else? All cells. Huh? Membrane. I don't know where the membrane is, but say membrane. Something else. Cytoplasm. Something else. Not all. No. So what? Ribosomes. That's it. Those 4 elements, all cells must have. Does that make sense? You cannot imagine the cell without cytoplasm, like empty inside, or without ribosomes, or without membrane, or without DNA. This is it. That's all cells have it. Is that clear? Just think about it. Now, can you throw something on top of the plasma membrane? Cell wall. Okay, some cells have cell wall. Can you throw something on top of cell wall? Capsule. It's generally called a glycocalyx. It can be present as capsule, slime layer. Do we have it? Yeah, like a capsule here. Okay. Going out there. Jeff mentioned flagellum, because some cells can move. Some cells have other attachments called pili and fimbriae. We'll chat about them. Okay, that's pretty much it. Some cells have inclusion bodies. Okay, here they store stuff in them. But if you begin with those 4 essential structures, if you just analyze what they do, you realize cell cannot survive without them. Does that make sense to you? Very good. Let's pause it.